Hey everyone, this is Jason, producer of Data and Biotech. I'm excited to announce that this podcast is now an AAPS media partner. Join us on site at PharmaSci 360, November 9th through to November 12th in vibrant San Antonio, Texas. We'll be interviewing leading scientists about the data trends that span the pharmaceutical development and manufacturing spectrum. Data and Biotech thanks AAPS for this opportunity to share emerging science with the global pharmaceutical community.
I myself and many people in this space are not thinking co-folding models will replace the physics-based methods, right? So ultimately, drug discovery is always a pipeline where you start broad and then you de-risk as part of the funnel. Okay, these co-folding models, they are breakthrough, they are really impressive, but today they don't yet have the impact on industrial research because the applicability for these industrial tasks is just not there yet.
Welcome to Data and Biotech, a podcast from Cordi where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. Here we go.
Robin Rohm, welcome to the Data and Biotech podcast.
Well, thank you for having me.
Awesome. Well, just to kick us off, would you mind giving us an introduction to your background and what brought you here today?
Sure. So I have a background in mathematics and medicine. And then I had a previous startup before Apheris in the genomics field, where we did essentially machine learning pipelines for secondary analysis. And so working with genomics data, that's very sensitive data, you always have that challenge of centralizing that data, bringing it to a point where you can analyze it. And so after going through that pain over and over again of not being able to move genomics data, we figured there must be a better way to use that data without moving it. And that was the founding story to Apheris.
Awesome. Will you give us an introduction to Apheris and how you put that sort of insight into practice?
Essentially, what you see in the industry when you look at how AI is coming to market is that you need very large and diverse data sets to train rich and powerful foundational models. And so essentially, for most of the, let's say, industry problems, that data is not out in the public domain. For text and maybe simple images, there is a vast amount of data just in the internet. But for sensitive data like health, life sciences data, molecular data, that data is missing. And so when we figured out how to essentially bring more diverse data that are owned by multiple organizations together, we decided to found Apheris with that mission, essentially help companies to unite their data sets without sharing it, utilizing a key technology that we pioneered, which is called federated computing.
Awesome. Well, we've talked a little bit on this podcast about federated computing previously. So but for those who didn't get a chance to listen to it previously, would you just introduce us to the concept of federated computing, maybe a little bit about how it works, and then how Apheris has sort of operationalized those ideas?
So essentially, an old paradigm when you are, let's say, applying AI to data is move data to a central location and then do all your modeling. That's the status quo still in many worlds. Now, there's a better paradigm to work with data that is owned by multiple parties. That is, instead of moving data to a central location, keep the data within the environments of the data custodians and bring models to that distributed data. So the key logic is, take a model, train small individualized models on each of the data custodians' own proprietary data, and then somehow aggregate these individual models to a global model. Reality is you need to iterate over that process, so it really requires an infrastructure to make that work, but that's the key concept. And so when we started working in that field, the first couple of years, we're really focused on making data owners, data custodians comfortable with that way of working. And as you can imagine, the key pillars here are governance, privacy, IP protection, and security. You're dealing with sensitive data, you're sending models around. So that have been the key pillars that we operationalized and productized over the years to make sure pharma companies can come together and collaborate.
Awesome. So, I know that one of the reasons we're here today is to talk about a specific collaboration that you have ongoing with regard to OpenFold 3. And so, would you just give us a little bit of, a history of that collaboration, how it came to be, and then maybe an introduction to OpenFold 3 and what makes it useful and valuable?
So first of all, maybe just introducing to kind of this use case, right? Like that's the field of co-folding. So what does co-folding mean? So essentially in drug discovery, what you're always interested in is the structure of the molecules that are interacting with each other. And the first real breakthrough here was AlphaFold 2 from DeepMind and isomorphic. Before ChatGPT, that was considered as the biggest AI breakthrough in history. And it solved this problem of, can I predict the structure of a protein based on sequence? And it turns out, yes, you can, as well with high confidence. Now, if you go to and look at drug discovery, the reality is they don't just need the structure of a protein. That's typically the target by which you search a molecule to interact with, right? You want to develop a molecule, a drug, that has a certain interaction with a target you're interested in to have a certain biological effect. And so this whole field is called co-folding. And the AI structural biology network, which is hosted on our product and facilitated by us, that's a network of large pharma companies where pharma companies come together, jointly utilize their proprietary data to train richer and more powerful co-folding models.
So obviously, if pharmaceutical companies are exploring, bringing their data into this federated computing environment that you have at Apheris, there's some value to making co-folding work. So if you could just orient us to what the value is to making co-folding work, and then why bringing these data assets together is going to help these companies to capture that value.
So maybe let's start with why co-folding is so hard to model. If you think about it, so modeling structure prediction as AlphaFold 2 is already a very hard problem. The benefit there is you have a lot of open data. The Protein Data Bank, PDB, is an open data repository with 240,000 structures. So a lot of model architecture innovation enabled DeepMind isomorphic to solve that problem. Now, if you take it to co-folding, two things change. Number one, the sheer number of combinations that you can model in co-folding, where you not just predict the structure of a protein, but as well how it interacts with other molecules, small molecules, large molecules, antibody, antigen interaction. So just that sheer number is orders of magnitude larger. And so you just have a much larger problem what you're trying to capture in a given model. The second, and I think more important problem here, is that there is not a rich enough data source in the public domain to meaningfully capture that phenomenon we are trying to solve. If you think about it, the PDB, right, 240,000 structures, but the number of protein-ligand interactions where the ligand has drug-like properties, right, so where it could be a drug, that is just 2% of that data repository. If you take out all the small molecules that are not interested, like sugars or so. And so suddenly you just end up with a few thousands of meaningful protein-ligand interactions in the public domain. Now, if you go to pharma companies, they have this data in their private data repositories. That's the key research that they did over the last couple of decades. However, as you can imagine, this data is incredibly sensitive, so opening it up or directly sharing it is not an option. And so that's where the AISB network, the AI Structural Biology Network, comes into play. We connect that data in an IP-preserving manner, hence get access to richer and more diverse data to capture that phenomenon we are trying to solve, which is co-folding.
Yeah, that's great. And before we move on to sort of like the structure of these models and how like federated learning for these models might work, can you just help us understand the relationship between AlphaFold 3 and OpenFold 3 and sort of like how they fit together or separately?
Sure. Let's start with AlphaFold 2 again. AlphaFold 2 is the DeepMind isomorphic model, and then there was OpenFold as the open source reproduction of that. And that again is for structure prediction. Then the next set of models here is, the first one was AlphaFold 3, which again is from DeepMind and isomorphic, which addressed that co-folding problem. And the challenge with AlphaFold 3 is that it's not commercially available and open source available. It is like there's an AlphaFold server, which is essentially AlphaFold 3, and you can query it, but only for academic purposes, for research purposes, and as well, it's hosted by DeepMind. So you need to share the query with a model that is hosted by them. So for industrial purposes, this model can't be used by pharma companies and internally fine-tuned to their data. Now, that is why the OpenFold consortium, which is a consortium where we are part of and many other pharma companies are part of, they fund Dr. Al-Quraishi's team from Columbia University to essentially build a complement of AlphaFold 3, fully open with all the engineering, such that you can fine-tune it, such that you can take it, locally host it. Dr. Al-Quraishi is an amazing researcher, and so that model is available to the parties of the OpenFold consortium right now, and it's it's getting fully open source release in a matter of weeks. So maybe by that time this podcast goes live, the model is out. And yeah, stay tuned. Impressive performance benchmarks.
Yeah, that's great. And so, one of the things I'm interested in understanding, since we have you on the podcast, is sort of, what were some of the structural model architecture or modeling insights that sort of gave us the leap from AlphaFold 2 to AlphaFold 3 or OpenFold 2 to OpenFold 3? And I think that'll lead sort of nicely into how the different data sources are being used inside of the modeling context.
Sure. So, I mean, we could go fully into the weeds here and speak about that. So I'll keep it at a certain level such that everyone can understand. But in a simple form, this model has an input derived from the sequence information, right? So the protein sequence. And then there's this big transformer model that essentially uses that. And then in the end, and I think that's the key aspect here of the co-folding models, there's this diffusion model, right now rather simple. What it essentially, this whole pipeline does, it uses a lot of additional information that you find in templates or MSAs. So MSAs are multiple sequence alignments, and that is essentially capturing evolutionary information that are sitting in other public data banks. So what you're essentially trying to do is, if you have a given protein sequence, can I get additional information about that protein family from that evolutionary information that helps me predict the structure of that given protein? And so that is all coupled in a pretty long data pipeline that this model then uses.
Yeah, so my understanding is that one of the things that allows this to be possible is the structure is much more flexible in AlphaFold 3, and it scales much better with data. It's relying on fewer assumptions about in the architecture itself. And so, as new data is added, the diffusion model adds several different aspects to the modeling outputs that make them really useful. One is that it's giving you sort of multiple outputs per sequence that you're getting, so that you could have multiple different options to choose from. And it's also giving you this sort of uncertainty quantification about the output that's allowing you to sort of prioritize the outputs and maybe utilize them more effectively. Am I understanding that correctly?
Yeah, in particular, this distillation approach, I think, is truly interesting, because as you say, what you're doing there is, during the learning, you're generating more examples and then taking these examples that the model then prioritizes as part of their learning process. And if you think about it, that's as well the area where just the rich pharma data repositories can generate a huge amount of value. Of course, you can add more structural data, but we might come to that as well. How can you add further data modalities to guide that distillation data process? And so things like binding data, right, that where you don't have the full structure, you can use to essentially guide what data during the distillation data process is generated. And this binding data is, of course, as well available in these pharma data repositories, which is very rich, very sensitive, but truly valuable for these type of model architectures.
Yeah, so this is one of the things that I was curious about as I look at the model architecture. Is my understanding is that as part of the multiple sequence alignment process at the beginning of the model, there's this sort of querying of the databases that produces sort of the relevant example sequences that are going to help to condition the model to, utilize the most relevant information from evolutionary history to produce the folding output that is most accurate based on the information that we have today. And so on some level, I understand that in federated learning, we're all sharing the weights, right? There's this latent space, these embeddings that get created that are sort of shared across these different data sources. But in terms of the OpenFold 3 model, it also seems like there's this initial conditioning process that is more like querying the raw examples before they get into the model. So I'm curious, how do you deal with that in a federated learning context that is sort of privacy protecting for the organizations that are sharing the sequences, but also producing the best possible outputs for the community that are then consuming the sequences?
Yeah, that's a great question. And by the way, there's multiple sequence alignment. You do not just need for the training, but as well for inference, right? As you query the model, you still need an MSA server to be connected. And the question is absolutely right. So how do you deal with that in a federated setup, both during training, but as well during inference as you use that model? What do you do as a company? So the reality is pharma companies have these MSA servers in-house, right? And so, but this is large data, and I mean there's often public data, but pharma companies as well have those enriched with their own templates and kind of their private ones. And so if you think about this federated computing architecture, but as well the, let's say, backbone that you need to use these models downstream, you see how much let's say technological innovation is really needed here, because we're not dealing here with a, let's say, simple, smaller model that is being trained. We're dealing with a full pipeline with querying of the MSA server. And so the rigor we've put into setting this up such that during training, but as well when you run these models on scale, no data, no private data or queries ever leave your local environments has been tremendous. There have been key innovations as well on the federated computing side that we did, where we worked with NVIDIA. So yeah, this is one of, to our knowledge, one of the, let's say, most complex and largest models that have ever been used in such a setting.
On the training side, how does the data enter the pipeline, and then what do you get in return? And then I'll be interested in the inference side as well.
Simplified the steps to get such a, let's say, setup up and running. We have a software component, it's called a gateway, that a pharma company can deploy within their own IT environments. And so they deploy this. Of course, you can imagine there's a lot of security validation that needs to happen before that happens. But once it is deployed, what a pharma company can do is register data. Now, register data just means you tell the gateway that data exists. It points to the storage location. So no movement of data, just if you're on AWS, on that S3 bucket, data that I want to use in a federated setting is sitting. The next thing pharma companies need to do, and that's very important, is set up access controls on a computational level. What does that mean? If you have a gateway, you point data that you want to use in a federated network. For pharma companies, of course, the question is, what are you using it for? Right? What computations will you run, and how do you ensure these computations protect the privacy and IP of my data? So this is something they can set up. They stay in the control. They say, this model that has been vetted with these security assessments and these privacy assessments, this model can run in these constraints. And so that is something the gateway then enforces. So if anyone tries to launch a computation that does not fulfill these settings, the computation is blocked. So those are the settings the custodian does. So each of the pharma companies do. And then once that is set up, every pharma company has their data in their own environments, the same settings. Then the model provider, in this case, Dr. Al-Quraishi, that we collaborate with the OpenFold 3 model, they are taking their model and sending it to the gateway. The gateway assesses, is this the right model? Is it the right configuration? Is it security vetted? And if yes, they deploy it and run it. And then all the data pipelines and all of that needs to happen. So everything we discussed before then happens via the gateway. And then these local models are aggregated over that process, is iterated until the federated model gets generated.
Okay. So if I'm understanding correctly, then, to answer my question from earlier, you're only accessing your own like MSA server, your own multiple sequence alignment server when you're working with the model, or when you're training the model. And then what gets shared back centrally is sort of like the changes to the weights of the model that are then that can then be used by other organizations. So like you're responsible for the data that conditions the training, and that later conditions the inference. But what's shared is really like the centralized weights, the embeddings, the that sort of thing in the model. Am I thinking about that right?
That's absolutely right. Only the weights, and only weights that are essentially assessed to not leak the underlying training data. So only those are shared. Of course, there's a lot of collaboration in such a network from a scientific perspective, and then, like alignment of how do we want to use these models? How do they get packaged? How do we do benchmarking? Like all of these things are, of course, aligned, but from a technical perspective, you're absolutely right. Only the weights are ultimately shared between the parties.
Yeah, that's really interesting. And sorry, before we move on, because I think it's just really relevant, which pharmaceutical companies, do you have working with you to do this federated learning right now?
So the ASB network is a large pharma to pharma network with 8 pharma companies involved. This specific project where we are training co-folding models in the small molecule field is with 5 pharma companies. That's AbbVie, J&J, Bristol Myers Squibb, Takeda, and Astex Pharmaceuticals. And all of them are contributing thousands of unique protein-ligand structures, contributing here, to be really clear, in a privacy-preserving manner, so not sharing any data.
Yeah, that's really interesting. So, obviously there's a lot of data. There's a lot of data moving in, and it's also past the legal, regulatory, and IP protecting hurdles of each of those organizations independently. So that's a strong vote of confidence in the federated learning framework that you're bringing to bear. One of the things that's interesting to me is you mentioned the limitations of the protein data bank that was used to train OpenFold 3, but I think I'd love to understand more about the limitations of the model outputs that result from like the model without this federated training component, and what's some of the limitations we expect to overcome by bringing in these other pharma companies' data sets into it.
Yeah, so that's a great question, and a lot of, let's say, the industry ecosystem is already looking at the limitations of these models based on benchmarks that have been built to, as well, let's say, visualize that in an intuitive manner. So maybe I can just quickly speak about the standard benchmarking technique here to assess these models' quality. So there's the runs and poses benchmark, and essentially, in a simplified manner, what it does is it is structured in different bins. So you have like different smaller validation sets, and these different bins or buckets, they are they have a different distance to the training data, which is the PDB. So in a nutshell, if you see that, you see on the left the data that is much further away from the training data from a similarity perspective. And then on the very right, you see the data that is very similar to what is captured in the PDB. And so when you look at all these co-folding models, AlphaFold 3, but as well the other open source equivalents or some private models that have been built, all co-folding models, you see the same trend and similar performance benchmarks, which is in the data that is similar to the PDB, these models perform quite high, often 90% of the structures they predict in the correct manner. But the further you go away, the less performant they become. And the performance drops significantly to something like 30 to 40%. Now, if you think about what pharma companies will be using this for, right? Like they will be applying these models to their own proprietary research programs with specific chemistry, with maybe targets that are not represented in the public domain, or at least with like chemistry that is unique. And so you will have a lot of problems that are much more on, like within these spins that are the harder ones. You will have few problems where pharma companies work on aspects that are really well represented in the PDB. And so that's a bit what the industry generally sees. Okay, these co-folding models, they are breakthrough, they are really impressive, but today they don't yet have the impact on industrial research because the applicability for these industrial tasks is just not there yet.
Yeah, that makes sense. And as I was researching this, it was like some of the things that came out as like being major limitations where it was insufficient data, and PDB was like any protein where there's like large conformational changes. So when like they're moving around a lot, like there isn't enough data to characterize that, or GPCR like it's biased toward like active states. It doesn't have sort of inactive or partially active or states in there when there's like 3 component systems, so like a ternary complex of three tripartite interactions. It's not working. I don't know if there's anything else that comes to mind for you, but those were some of the things that that came out.
No, I think those are like some of the key challenges. I mean, there's like aspects like stereochemistry that is still a problem. And to be as well clear here, right? So proprietary data that is rich and diverse is certainly a key pillar to respond to that. But of course, there's as well model architectural innovation that can help with that, right? And so I think a key aspect with these foundational models is that if you don't build the foundation right, all the downstream applications as well don't become really well. And I think the binding affinity is an interesting example of that. So there is this, essentially binding affinity is a huge problem. And for lead optimization or virtual screening, you want kind of the ability to predict binding affinity early in the process. And so the key idea here is if you predict the structure of a protein-ligand interaction really well, can you not just use that as an input for a binding affinity head that then predicts you the binding affinity? And turns out, yes, that is something that you can build, and it works reasonably well if the structure prediction is correct. So that's why you can post great academic papers that show exactly this. But the reality is, of course, if the structure prediction is not correct, why would this binding affinity head as a downstream application be correct, right? It wouldn't make any sense. And of course, that's as well what we see in the data. And so that's why you need to invest in that foundation really well. You need to get a good, high quality structure prediction across the different data modalities you're looking at and the diverse chemistry, such that all these downstream applications for lead optimization, virtual screening become as well useful for industrial applications.
Yeah, that makes a lot of sense. And also, one of the things that I was reading was that predicting binding affinity is something that you should sort of do in order to evaluate the results that come out of a model like OpenFold 3. You'll use these very high computation physics-based models to do that kind of prediction, but there's sort of this hope that if you bring all of these diverse data sets together and create that better foundation that you're describing and, more accurately characterize the structure of these proteins, then the binding affinity can be predicted accurately with, much lower computational overhead, which allows for much better, in silico creation and evaluation of these different proteins. Am I thinking about that right?
Absolutely. That's the hope. And to be clear, right? So I think I myself and many people in this space are not thinking co-folding models will replace the physics-based methods, right? So ultimately, drug discovery is always a pipeline where you start broad and then you de-risk as part of the funnel. And of course, as well, data generation with labs to confirm what you're seeing is a key pillar around that. So what you're trying to do is accelerate and lower the costs and increase the timelines of how fast can you iterate over that space. And so if you have a method that with much higher likelihood and much shorter timelines as a co-folding model that predicts structure, if you can use that earlier, you can use it for things like lead optimization or virtual screening use cases where you can go over a much broader space of chemistry compared to a physics-based method where you would use half a day to compute such a structure, right? So the number of examples you can just compute is just much slower than compared to a co-folding model, if it works correctly, of course.
Yeah, that makes a lot of sense. So, as the different partners are bringing their data sets to Apheris and to, federated learning for OpenFold 3, are you or are they working together to, apply like automated data quality or curation approaches to the data to make sure that, collectively they're all lifting the quality of the model versus, one partner maybe has different data quality practices than another. And so, it leads to like a worse model for everyone else.
So I think let's say, fair data contributions and high quality data contributions are the key biggest reasons why federated networks succeed or fail. So yes, there's a lot of rigor exactly around this aspect. Now, the good thing with structural data compared to some other data modalities in the molecular field is that it is reasonably well structured. Now, still, the reality of that is still there's a lot of nuances of how you can label that data. So there still needs to be a lot of rigor to that. And so, yes, we have essentially controls, both guidelines and controls. So it's about enablement, but as well about qualifying that before data is fed into a given model, you check the quality. But at the same time, you as well want to provide the scripts and functionality to take data that pharma companies today have to map it to what you need. And all of that is packaged essentially in the products that we've built, which goes beyond a federated computing infrastructure, right? This is additionally scripts that take structural data and transform it and then validate it and then feed it into the model. So that's a lot of extra product functionality that we've built to enable such use cases.
You mentioned sort of the benchmarking approaches and how, AlphaFold 3 and OpenFold 3 are evaluated. Do you have benchmarks or metrics that are showing sort of like the contribution that that this federated approach is making toward making OpenFold 3 a better model?
So yes, but I can't kind of speak about the numbers here. Of course, there's a lot of people with, let's say, stake that want to make sure this gets into the right publication. So of course, people will write about this. So I can't speak about the details here. But of course, all of that will be published. And I think what is interesting, maybe what I can speak about is the rigorous benchmarking you want to do here, right? So benchmarking in a federated manner is a true complexity because you can't see the data. And so splitting the data such that you can assess aspects like generalizability is really hard. And so what we've done here is essentially making sure the runs and poses difficulty is retained. So you take pharma data and you ensure that the training data you feed in does not leak any information that you have in the runs and poses data set, such that if you evaluate it in runs and poses, you can have that true comparison. So that's one benchmarking approach. There's another one to just hold out private data to see how good does it work on programs the data has never seen. But both of those are used in the benchmarking strategy.
That makes a lot of sense. Is there a thought that in the future, like the federated, fine-tuned version of this model will be made open source or publicly available, or how is the AISB thinking about that?
So the AISB network is structured such that there is additional incentives for further parties to join that network, and one of the key incentives is, of course, the, let's say, give and get of such a network. The give is you provide access to data, proprietary data that you own, and the get is you get access to these models that have been trained on the richer and more diverse data and can then use them downstream for your own drug research programs. And so there is this ability to join the AISB network if you bring data, and in order to keep that up, of course, you don't want to have these models openly available. Next to that, and I do want to say that there's, of course, as well a very rigorous security and IP protection posture. And that is one that has a lot of technical safeguards, but as well legal and organizational ones. And so the threat model changes, of course, as well, the more open you go. And so that's another reason why to date, these models are only accessible to the parties that as well participated in this network.
Yeah, that makes sense. And how, if there's an organization out there that's interested in joining the network, what do they like? Can you give some idea of like what they need to bring to the table and how they go about joining?
Sure. So they need to bring structural data, and potentially in the future, as we launch new initiatives, binding data to the table, proprietary data. Data contributions may differ. There is, let's say, models that you can think of that as well allow smaller companies to join. Right now, this is mostly for the largest pharma companies. But indeed, you can think of network setups where as well you can allow smaller pharma companies to participate and have a fair data exchange. But of course, as you can imagine, with such a large, let's say, names as these 8 very large pharma companies, there's a lot of governance as well around it. And so the key process is you reach out to us and we help you get into the network. And the network has an overarching contract that allows you to be part of it. And then based on that, the data contribution requirements and the give and get is defined.
Awesome. So, when we were talking earlier, you talked about like the sort of the adoption of these models that are coming out of that are like trained using federated computing approaches. And, maybe it's not just as simple as computational chemists and drug discovery teams like picking up this federated trained model off the shelf and then using it in their day-to-day work. So I'm just interested in, what have you seen in terms of adoption from pharma companies that want to use these models? Like, what are some of the barriers? And then, if there's anything that you've seen that has been best practice, would love to know that too.
To be clear, I think that's one of the biggest learnings that we made over the let's say years of supporting this is, okay, if you get to a federated model and now you have that federated model, you can even benchmark it and show that it's superior. That is still a big gap for bringing it to users who are then using it in real world drug discovery programs. And so these co-founding models, who are they for? Medicinal chemists and computational chemists. These are slightly different user groups. And in a large pharma context as well, typically these user groups work together on drug research programs. So what we've developed in the course of that is essentially a product, we call it AlphaFold, and it's accessible as well to parties that are not part of these federated networks. Then you don't get the federated models, but just the open ones that are trained on public data. But what it essentially allows you to do as a pharma company is take these models, locally deploy them, have an interface for both computational chemists and medicinal chemists jointly in one interface, and then start with benchmarking and assessing the applicability domain on your private data. So benchmarking, that's a more rigorous approach where you need to build the right benchmarking data set and do that in a proper manner. But sometimes a medicinal chemist as well wants to assess the applicability by just looking at a structure in a visualizer, right? So both is available in that tool. And so what happens is typically these parties start with benchmarking and assessing applicability, but then quickly move to the question of, okay, how can I now customize that and improve that for my specific drug research program? And that can have various forms. That can be a fine-tuning workflow. That can be additional templates where you tell the model, predict something in that area. That can be just scaling it out, right? Like instead of running single predictions, run like thousands and prioritize that and use the output to filter and do that. And so all of that is available in a tool that we provide. And again, that is a very easy way to start for smaller companies that want to get let's say in the game of co-folding. That's something that we built for the product community.
Interesting. And another thing that we talked about previously was this idea that once you start this process of federated learning for improving or fine tuning a model like OpenFold 3. you can start to assess where the gaps are in the data set that's feeding that model, and that there might be a role for data generators like CROs and high throughput labs to play in strengthening the model. So I'm just interested in hearing from you, how do you assess those gaps? And then, what is the role that you see for these data generators in, improving the models in the future?
So first of all, like quickly explaining the key idea here. And so I think generally, if you have a federated network, right, you have a set of parties, and these set of parties, of course, bring much richer and more diverse data than typically what any individual company or the public domain has, right? So that's a huge step forward. However, to really solve a problem like co-folding or different problems, ADME predictions, antibody developability, you name it, right? Like these hard industry problems. Of course, very often, even the data in a federated network is not comprehensive and captures the whole space in a meaningful manner. So what you can essentially do is, based on the model's behaviors, based on intelligent benchmarking, diversity assessments, confidence assessments, you get a signal of where is the model already strong and where do you have gaps in that existing data network, that if you could complement that, you would meaningfully increase the value for all network participants. And so that's something that you can do. It follows the logic of what you typically do in an active learning program, right? Where you use essentially a model to guide the next data that you generate. That's something similar from a technology perspective, what you can do here. And so you can work with a data generator that then fills these gaps in a systematic manner. We are pioneering that in a different network setup together with Ginkgo Data Points. Ginkgo Bioworks is the mother, where it's the field of antibody developability. So this is a different model category, but that's for the first time where historic data and then new data that is generated by Ginkgo in a high throughput manner is used to address antibody developability. And I think that combination is really powerful because it addresses exactly the problem of bring parties together to use what they historically have, but as well be intelligent and thoughtful of what new data to generate in a cost-effective manner. So that's what I'm really excited about, and what I expect to see much more in the networks that we power, and generally the industry networks that are out there.
Yeah, and that makes me think that, sort of the opportunities for federated learning in the biotech and life sciences and healthcare space is sort of unlimited. There's a lot of these problems have similar structures where, the most important data is housed inside of these private organizations that would all benefit from, modeling together, but, also are very protective of their IP. So I'm just interested in, whether you see a future where these federated networks can span not just, pharma, but, also regulators or biotech startups or hospital systems, or, all the different groups that could contribute data to an effort like this.
Totally. So I think there are these large, important industry problems. Co-folding is one of them, but there's, of course, like others in the early stage drug discovery stages. But of course, as you move down towards clinical data, predicting clinical success, in vivo PK prediction, new data modalities, like there's the whole field of omics and single cell data that is used. And I think all of them follow a similar pattern, right? That data is generated by different parties. It's sensitive. If you move to clinical data, it's well from a regulatory perspective and a patient privacy perspective, truly sensitive. And as well for all of those, let's say, use cases, what is true is there is a lot of existing data. But even if you could combine all of that existing data, the problem that you're modeling in biology is still so complex that you need additional data to generate, and that data generation is expensive. So if you could lower the costs for a set of partners to generate that data, that would be truly valuable. And so I think the key answer to many of these problems will be federated networks coupled to targeted data generation to lower the cost for each individual participant and have joint ambitious projects. And so yeah, if you look at our portfolio, right, we have a couple of these networks that we host ourselves, ADME, antibody developability, co-folding, but there are a couple of more that we will be hosting. But as well, our customers are partly building their own networks. So for example, pharma to site networks is a key thing that our customers build, and we just provide the technology, but then they select the set of sites that they build a network to do clinical research, patient demographics, and things like that. So I think a lot of new innovation will happen in exactly that field.
Very interesting. And, going back to the co-folding problem that we spent most of the episode on, do you have a sense of how close or how far away we are from, co-folding models that are, as impactful for on co-folding as AlphaFold 2 was for single protein structure prediction, or how close we are to having co-folding models that are really driving, usable binding affinity calculations that can be used downstream and filtering the stuff down.
So we call that the AlphaFold 2 moment for co-folding, right? I think that would be truly impactful for the industry. How close are we? I think the key ingredients are there, but you need to bring them together. And of course, there's still scientific innovation that needs to happen here. So key pillars: industrial data at scale, right? That's a key pillar. And I think with the AISB network, we are doing exactly this. The second key thing will be architectural innovation. And I'm truly excited about the OpenFold ecosystem because they have now built this model. It's open source, it's soon open source available, and it has this modular engineering pipeline. So what you will see is many parties will be building upon that. And so that, I think, will allow for a lot of academic groups to further take this further. Dr. Al-Quraishi will be a key thought leader here, but as well potentially companies packaging this and applying it to different aspects. So architectural innovation are going to be key. And then the last point that I think is critical is targeted data generation. So there are a couple of initiatives out there. I do think that we will see both large initiatives generating more public data, but as well more focused targeted data generation that pharma companies will do, right? So they have a certain area that they're interested in. And so today they are already generating data for that. So how can you generate that data such that you can use it to fine tune these models as you do your drug research? That's, I think, a truly interesting field where we are as well playing with our applications to make sure the moment new data comes in, parties can fine tune and customize that to their drug research programs to get the most out of it for their drug research program. So all of that combined, but I think it'll take some more time to really get this together.
Yeah, it sounds like we're closer than we've ever been, but still have a lot of work to do. So, we've touched on a lot of aspects of federated computing, of co-folding, of sort of how these networks are formed. Is there, if you had one takeaway that you wanted the audience to take from this conversation, what would it be?
So I would say co-folding is coming, even if it's not perfect today for your specific research domain. It is coming, and if something like that is coming, you don't want to run away from it, right? You want to run towards it. That's the same with AI. AI won't solve all your operational problems, but you certainly want to embrace it. You want to get used to it. You want to set it up. You want to test it. And I think the value here is you can start small, right? Not every company needs to be part of these large networks from the start. Whether it's a large or a small company, you can deploy these models locally. They are openly available. And so I think it's really important that pharma companies, as well as smaller pharma companies, don't forget the security angle here. So deploy them locally, use them. And if you have any challenges around that, reach out to us. We are happy to help and make that as streamlined and easy as possible for you.
Awesome. And where can people go to learn more about Apheris and the AISB network?
So Apheris.com, there's a large tab on the AISB networks and some other networks that we host on the website, so you can directly go there. As well, of course, LinkedIn, Robin Rohm, you can reach out to me if you're interested. So those are probably the best entry points.
Fantastic. Well, Robin, it's been great to have you on the podcast. Really appreciate the time and look forward to connecting down the line.
Awesome. Thank you for having me.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.